All right, let's talk about exactly how the mammalian ear works, and then we'll talk about vision and a little bit about smell and taste as well in this video. So our ear has an outer ear, a middle ear, and an inner ear, and it's important to understand the distinction between these three areas. So the outer ear, another way to think of it is where you can stick a Q-tip. That's the outer ear. Everything outside of the eardrum, which is also called the tympanic membrane. The middle ear is generally what gets infected if you have an ear infection. If you have a middle ear infection, it means that this area here is full of fluid generally. And this can often happen when you have a sinus infection or a cold, because the middle ear is connected to your throat by the eustachian tube. And in the middle ear, which normally should not have any fluid in it if you don't have an ear infection, is how the sound gets to the inner ear, which is where the neurons are connected to. So the middle ear has three little bones, the malleus, the incus, and the stapes. Malleus means hammer, incus means anvil in Latin, and stapes is from the Latin word for stirrup, like what you would have attached to the saddle of a horse to put your foot in. That's a stirrup. So malleus, incus, and stapes. And the stapes is going to vibrate on this little window here, which they don't have an indicator here because you can't really see it, the oval window. And that's going to vibrate the fluid in the cochlea. And the round window here dampens the sound. So sound goes this way by this stapes vibrating here on the oval window. It goes into the cochlea, and then it comes back around, and the round window acts like a dampener to that sound. And those are parts of the inner ear. So everything after the little oval window and the round window here, that's all the inner ear, where we have the semicircular canals, which are part of our system of balance and proprioception, and then the cochlea, which is how we hear sound. And this is a little, let me go back here. So see this little square here? We're going to take a slice through the cochlea. It's a coiled up, but I'll show you some images of it more stretched out so that we can kind of see the structures in there. But if we look at a little slice in here in the cochlea, so we have two fluid-filled canals, the vestibular canal and the tympanic canal. And that's where the sound pressure wave is going to start from. One has the oval window at the end of it, and one has the round window at the end of it. And then the cochlear duct. And these are all fluid filled. And this little region in the center here where the ciliated cells are that are connected to the neurons that are actually going to take the signal to the brain, this little region in the middle here is called the organ of Corti. And since we capitalize the C here, that's an indication that it's named after the person who first described it and discovered it. I think cochlea has something to do with the snail for the shape. That's not a person's name. So if we take a closer look now at the organ of Corti, so this is where the hearing actually occurs. And also if you damage your hearing with loud noise, this is where the damage occurs as well. So the basilar membrane here is going to get deformed by fluid pressure waves in the fluid that's being vibrated by the stapes hitting the oval window. The basilar membrane will vibrate, and that's going to cause the hair, well, they say hairs, but they're actually cilia on these sensory neurons. They're going to be moved by being bumped up and down against the tectoral membrane, which is pretty stiff. And then that signal is what is going to go to the auditory nerve. And the organ of Corti can detect both the amplitude of sound, like how loud it is, and the pitch. And that's because different areas of the basilar membrane are different thicknesses, and that's going to and they're only going to vibrate to certain frequencies of sound. If we take a close-up look at what those little hairs look like, so this is one cell here, one hair cell, and keep in mind that when I say hair, they're actually cilia. So if you remember back from last semester, what is the definition of a cilia? That means it has microtubules inside, it's covered with a plasma membrane. So these are not microvilli, because microvilli have actin filaments inside. Cilia have microtubules inside of them, although they can look really similar under an electron microscope. And these are non-motile cilia because they can't move like the cilia in the fallopian tubes or in your lungs, for example. These cilia are non-motile, but they get moved by bumping up against that membrane when sound bends the basilar membrane. So the first thing that happens is the tympanic membrane, your eardrum, vibrates. And that's going to vibrate the malleus and the incus and the stapes, and the end of the stapes here is going to vibrate up against the oval window. And that's going to vibrate or create a pressure wave in the fluid that's inside the cochlea. So here is the cochlea kind of unrolled a little bit so that we can see it better. So here's the stapes in the oval window. The stapes vibrates the oval window, and that's going to create a fluid pressure wave here, and that is what is going to deflect. This pressure wave is going to deflect some portion of the basilar membrane depending on the frequency or the pitch of that particular sound. And that's how it's going to be. And then if that is depressed, wherever portion, depending on the pitch, how high or low it is, that's going to indicate where along the basilar membrane we're going to get that pressure wave deflecting the basilar membrane. That's going to push those little cilia up against that stiff membrane, and that's going to create the signal. That's going to create an action potential. So here we have our hair cells, and you notice they actually put hair in quotes because they're not really hairs, they're cilia. And that is going to release neurotransmitter in response to the hairs being deflected in one direction but not the other direction. So bending of the hairs, if the hairs are bent a lot, that's going to release a lot of neurotransmitter, and that's going to generate a signal. Now this is one of the reasons for tinnitus. When people hear constant ringing in their ears of a particular pitch, one of the hypotheses is that this is caused by damage to these hair cells from loud noise at a particular spot in the basilar membrane, such that neurotransmitter is always being released at some moderate level, and that creates the perception of sound. Because remember, the brain is interpreting signals based on which neurons those signals come from. So if the neuron comes from a hair cell that is supposed to be hearing high-pitched noises and it's damaged, and instead it's releasing neurotransmitter all the time, that's going to be perceived by the brain as a high-pitched sound that doesn't stop. Tinnitus can also come and go depending on if you have pressure in your ears for some other reason, like you have a sinus infection. There's other weird medical conditions that can also temporarily cause tinnitus because you're changing the signal that's being sent, and that's an indication. Your brain is receiving action potentials from specific neurons in your ear when you have tinnitus, and it might represent actual sound, or it might be because of damage, either temporary or permanent damage to those hair cells. So volume seems obvious how we would perceive volume. If a sound is louder, there's going to be more action potentials, and the sound will be perceived as louder. Pitch, though, how do we perceive pitch, different pitches? And this is something that almost all humans can easily do. We can tell if a sound is higher or lower than the sound we just heard before, and we can learn scales and other organizations of different pitches. So how do we do that? That has to do with the basilar membrane. So the basilar membrane, so this is the cochlea unrolled. This is the organ of that membrane inside the organ of Corti, completely unrolled into a straight line. So it's all curled up, but it's not the same width. It's not the same, it's not uniform. It's thicker in some areas than in others, and so it's not going to vibrate to the same frequency as easily. So the thinner areas of the basilar membrane vibrate to lower frequencies. The thicker, fatter areas of the basilar membrane vibrate to higher pitch frequencies. So that's why you can lose certain pitches but not others, and we tend to lose the higher pitches first. And that's very common just with aging. People tend to lose the ability to hear high-pitched sounds, but not low-pitched sounds. And this just shows that we have different areas. If there's a particular sound, only that's a single pitch, only one area of the basilar membrane is going to respond and vibrate best to that specific pitch, and that's how we can distinguish different pitches. So sound waves are coming in first through the vestibular canal and then back out through the tympanic canal and then dampened at the round window. So our equilibrium doesn't use the cochlea, but it uses a nearby connected area. And there's really two things. There's a sensation of gravity that comes from the utricle and the saccule, which are closer to the cochlea. And then there are the semicircular canals. So, and that's how we detect motion of our head, not anything else. And we detect gravity with the utricle and the saccule. And there are, again, otoliths, very similar to the way that insects detect gravity and plants detect gravity. Only this time they're not going to flop around in a liquid. They're kind of embedded in a gel inside of here. But if you're upright or laying down or upside down, these little liths are going to move a little bit and touch the hair cells again, and that gives you an indication of which direction is up according to gravity. And then we have the three semicircular canals, which are filled with fluid, and the fluid in there will move, and they point in different directions. The three semicircular canals are in different orientations so that you can detect movement of your head in any direction because it will move the fluid in those canals. And this, instead of using otoliths, there's this gelatinous layer on top of hair cells, and fluid that's flowing in the semicircular canals causes this little blob of gel to move, and that bends the cilia, and again, not really hairs, cilia. It bends the cilia, and that indicates, depending on which way the fluid is moving, that tells us which way our head is moving. And if something is wrong with the semicircular canals, like let's say you have a viral infection and pressure builds up in the semicircular canals so that they're not moving correctly, they don't move correctly with fluid, you can feel dizzy, you can get vertigo where you feel like the room is spinning. So that's what happens when those go wrong. Occasionally people completely lose the ability to even stand up because they have totally lost the ability to tell up from down. Usually that's more serious than just an ear infection. Okay, so for vision, most animals can detect light. And there's a difference between just being able to detect light and tell light from dark and having eyes that can see shapes or focus. There's all different degrees of eyes. The simplest eye is just a little spot of photoreceptor cells that can simply tell it's dark or it's light. And the most complex kind of eyes can form focused images in the brain, and we're not the only animals that have that. And vertebrates aren't the only animals that have that ability to see clear, focused images and have depth perception. Insects, or some arachnids, some invertebrates like cephalopods, this is an octopus here, have that ability, have complex eyes that can focus clear images. But many animals do not. Many animals can only detect, is it day, is it night? And even the simplest animals, well, I don't know about sponges specifically, but cnidarians like jellyfish have little photoreceptor cells, and they can definitely tell, is it light or is it dark? So, and interestingly, in all animals, now that we can study the genetics of photoreception and photoreceptors and vision, some of the same genes are involved with producing whatever kind of vision an animal has across the entire animal kingdom, which tells us that these genes are ancestral to the animal kingdom. And although eyes vary quite a bit, and how complex that photoreception is varies quite a bit, some of the basis of that is consistent. There are some controlling genes like PAX2 that controls the expression of a lot of other genes that's involved in vision in all animals that have vision. And some of the photoreceptor pigments as well are common across a wide spectrum of animals. The simplest kind of eye that animals have are simply eye spots that don't focus any image. All they can tell is it light, is it dark? And flatworms have eyes like this. They also can use them for detecting predators. So if it's light out and suddenly it gets dark, that tells them that possibly a predator has just moved overhead. And many animals that have this type of eye take action when it suddenly gets dark. They will go back into their shell, or if it's a bivalve, it will close its shell if it suddenly gets dark. So they use these for more things than just telling day from light, but it seems like being able to tell whether it's day or night and having a circadian rhythm has certainly been important in the animal kingdom. So planarians and other organisms with simple eyes like this. In the case of planarians, they're using it to avoid light. They tend to, in freshwater especially, they live in the mud at the bottom of ponds. They don't want to be in bright light, but they can't see any images. All they can tell is the light getting brighter? I keep moving this way, the light is getting brighter. They'll turn and go the other way. The light's getting darker now? Okay, that's where I want to go. I'll keep going that way. That's about the limit of their information, and they can tell if it's nighttime or daytime. Insects and crustaceans, if you remember, these are actually quite closely related. Within the arthropods, insects and crustaceans are in the same order within arthropods now. And that kind of makes sense. They have the best vision among arthropods. Crustaceans, including shrimp and lobsters, especially certain kinds of shrimp, have really good vision because they're predators. And that's kind of a trend among animals, is that the best vision is often among organisms that are predators, or in the case of insects, organisms that need really good 3D depth perception. When the case of flying animals need that. And they have, insects and crustaceans don't have single eyes, they have compound eyes that are actually made up of hundreds to thousands of tiny individual eyes that each send an individual image to their brain, that the brain then puts together into a single image. And each one of their individual little eyes is called an ommatidia. And so that's why they're called compound eyes. And compound eyes are extremely good eyes. They have very good vision. It used to be thought, of course, that since they don't have eyes like us, that they didn't have very good vision. But lab experiments have shown that it's not true. They have really good color vision. Some insects like bees and certain butterflies can even see in the ultraviolet, which we can't. And this is what one of those ommatidia, well, the singular's ommatidium, because of course Latin, what they look like. And it's, in some ways, it's roughly resembles one of our eyes in that they have a transparent layer, a cornea. They have many fewer cells than are present in one of our eyes. They have photoreceptor cells that connect to neurons that relay that image to the brain. Single lens eyes are found in some arthropods as well, like spiders have single lens eyes, not compound eyes. But also mollusks like octopus have single lens eyes, which very much resemble our eyes, except they evolved completely independently. Single lens eyes often have, not always, but often, especially invertebrate eyes, they have both a way to change the amount of light that's coming into the eye. So that would be the iris. That's the colored part of the eye that we can see on our eyes and also on this owl. The iris is muscular, and so it can open to allow more light in and close to allow less light in. The pupil is that opening in the iris. There's also a lens that can accommodate for distance. So not all animals have a lens that can accommodate for distance. So we can focus on things far away, and then things that are close up, that's because of our lens in our eye, not the iris. The iris changes the amount of light that enters your eye. We have eyes that can detect color as well, and that is, we detect both the amount of light in striking a specific cell in our retina, and some of our photoreceptors can also respond to the color of that light, but not all of our photoreceptors, and different animals vary in how good their color vision is. Insects and crustaceans have really good color vision. Other animals have extremely poor color vision. We have pretty good color vision. Primates have pretty good color vision. So here's what human eye looks like. So light's coming in this way. So cornea is the clear layer. That's what you're putting a contact lens on. That's your cornea. The iris is, we can see through the cornea. That's what is, it's muscular, and that is what's opening and closing in response to light. The lens is also clear, and the lens is made up of protein, and it stiffens up with age and also with exposure to ultraviolet light. So if you're out in the sun a lot during your life, the lens, that's going to damage the protein in the lens, and it will stiffen up. And what happens when it stiffens up is you lose the ability to accommodate. So as you get older, usually the first thing that's noticeable is you can't read anymore. You can't focus on things that are close up, and you end up holding things really far away to try to read them. But eventually, as that lens stiffens up, you also lose the ability to accommodate for distance as well, which is why sometimes people, they never wear glasses their entire life, and then eventually they need reading glasses, and then finally they need glasses for distance and for reading because their lens is stiffening up. There's a vitreous gel or vitreous humor inside the eyeball that's transparent. If you have floaters, this is where your floaters are. Floaters are little solid chunks that are not transparent that end up in your vitreous gel after they're just debris. They break off. They're not cells. They're little chunks of protein and stuff. And sometimes you can see them, like looking through a microscope, sometimes you can see your floaters, or looking at a blank white wall, sometimes you can see your floaters. They don't ever go away. Sometimes they get worse as you get older. The retina is where the photoreceptor cells are, and there's also a pigmented layer called the choroid. The rods and the cones that you've heard about, these are the actual photoreceptors. So the rods only detect that there is light. The cones can detect what color of the spectrum there is. The optical disc is where the neurons all dive through the retina to go to the brain, and so that's actually a blind spot. There aren't any rods or cones in the optical disc. There is an area where we have our highest fine detail vision, the fovea. Fovea and optic disc are sometimes confused, but they're actually kind of opposites. The optic disc is a blind spot. The fovea is an area of really high detail vision. That's what you use to read, is your fovea. So here's the same information. So the choroid is a pigmented layer. The retina is the layer of neurons. The optic disc is where all those neurons from the retina dive through to go to the optic nerve, and so there's not actually any rods and cones there. And then the fovea is our area of really our best vision in, well, we need adequate light for it because cones need more light than rods. And this is a nice image. You can kind of see how the iris is muscular. And the lens is attached to muscles, and the lens, the muscles around the lens can either squish it and make it smaller, which is how you see close up, or they pull on it and they stretch it out so that you can see things further away and focus on them. A close-up of the retina here. So the neurons are in front of the photoreceptor. So light is coming from over here on the left, and light goes this way, and the photoreceptors send their signal to, there are some neurons in between the photoreceptors and the optic nerve neurons, and these neurons actually do a little bit of processing of the information. They are attached to one another, and there is a little bit of visual processing that occurs before the signal ever gets to your brain, which is these neurons in between here. They're the reason why there are so many optical illusions that you've probably seen, where people show you things where, you know, a line will disappear or something. That's why, because these neurons are, they're, most of the time what they're doing is aiding our vision and making our vision better by enhancing contrast between things that aren't very contrasty. But we can reveal the problems with them with these various optical illusions. So the rods, we have more rods. Rods are really sensitive to light, but they don't give us very much detail, and they don't distinguish color at all. Cones give us better fine detail and also can distinguish color, but they require a lot more light to be activated. So that's why if you're in dim light, you can't see color. And probably all of us have had the experience of like putting socks on or something in a dim room, and then you find out later that you put on socks that were two different colors. That's why. It's because if you're in low light conditions, your cones are not receiving enough light to be activated. So you can't see color, although your brain will fill it in, and your brain will tell you that it's seeing color, and it's actually not, which is why it's very easy to put on things that are too, I've even had the experience of having shoes that are exactly the same shoe, but they're slightly different colors like brown and black, and I've put on two different color shoes in a dim room because in the dimness they look the same. So test yourself if you don't believe me. In dim light, cones aren't active, which is also why it's hard to read in dim light. So we need our cones to see fine detail and read, print. So if the light gets too dim, you can see that there's print there, but you can't read it because your fovea that we use for reading needs the cones to work, or it doesn't work. And vision is interesting in that it's an inhibitory system. So when it's dark, there is neurotransmitter being released, and when it's light, neurotransmitter stops being released. So it's kind of the opposite of what you think. And our visual cortex is similar. So the way that visual information goes to our brain, we have, if you consider the field of view to have a right half and a left half, if you remember from our motor section that your right half of your brain controls the left half of your body, and your right hand is being controlled by the left half of your brain. So your brain also wants to get information. The left half of your brain wants to get information from the right visual field. And so the information from both eyes is actually going to one side or the other side because some of that information is crossing over. So something on the right, something on the left. It's going to be seen by both eyes, but because the neurons cross over, all the information from your right visual field is going to be processed in the left side of your brain, and from your left visual field is going to go to the right side of your brain. So it's an interesting process that goes on with vision. Color vision, if you're colorblind, about 7% of humans are colorblind, and green-red colorblindness, I should say. There's very rare types of complete colorblindness, individuals who have only one type of cone. Instead, we have 3 types of cones. So people who are red-green colorblind are missing one kind of cone. So to them, red and green look the same. That's the most common kind of color blindness because that gene happens to be on the X chromosome and males only have one. So of the 7% of humans that are colorblind, almost all of them are male because of that. There are other more rare kinds of color blindness that are on the autosomal chromosomes as well. So if you've never been tested, probably everybody under 30 has been tested in grade school for color blindness. So if you are colorblind, you won't be able to see the numbers inside of these bubbles here. You should be able to faintly see a number inside each one of those. But if you're colorblind, don't worry about it. It's not even considered a genetic disorder. It's more of a quirk. It may have evolved by natural selection. Individuals that are colorblind have slightly better night vision than people who with normal color vision. It may have been something that was actually a positive during our hunter-gatherer past. Other primates don't have as good a color vision as apes do, including us. Like we have 3 different kinds of cones that can see red or green or blue. Some monkeys only have 2 kinds of cones, and so they can't distinguish as many colors as we do. So the most detail is found is received by the fovea in the retina, which is why when you look out in your peripheral vision, you can't see things with as much detail. Only in the very center of your visual field do you have the precise detail. And that's the fovea, and it's almost all rods or almost all cones, very few rods, which is why we have trouble. And remember, the cones need high light to work. They're not even activated in really low light, which is why it's really hard to read or see fine details of things if the light is too dim. And it also requires that your lens be able to accommodate for whatever distance there is. You're not going to get a clear image on your fovea if your lens is not accommodating correctly, which is why so many of us need glasses to help with that. So this is how accommodation works. So here's the lens. And for near vision, those muscles contract and squish it into a short and round, thick shape. And that lets us focus on close things. For middle vision, the muscles are all relaxed. And then for distance vision, the muscles pull on the lens and stretch it out flat, and that helps us focus on things far away. And it's not unusual at all for people to have problems with one of these, even as a young person. Having trouble with distance is by far the more common. Being nearsighted, that's called, means you have near vision. If you're nearsighted, you have near vision. You can focus on things that are close up, but you can't focus on things that are far away. If you're farsighted, that means you can't focus on things close up. But you can focus on things that are far away. That's much less common in young people, especially. Okay, let's talk about taste and smell. So taste is what your tongue and your mouth does. Smell is what your nose does. And for things that live in the water, both of those things would be the same. But for land animals, those two senses are different. So for gustation or taste, these are things that are dissolved in water or in our food. It's whatever we're putting in our mouth or drinking, for drinking water. It's molecules that are dissolved. That's what's going to be detected. Smell is things that are in the air. And although we tend to put these two things together, because when you eat food, some of the molecules go up into your nose and we are actually smelling them. We think we're tasting them, but we're actually smelling them with our nose. As you've all found out, if you hold your nose, or if you lose your sense of smell because you have a cold, or hopefully not COVID, you can still taste, but you can't smell anything anymore. So food loses most of its what we call taste. We say we can't taste or smell our food, but really the tongue is unaffected, and we're still going to be able to taste the things that can be tasted. So our tongue has 5 tastes that we can perceive. So these are not smell. These are things that are perceived by the tongue and a few other places in your mouth, and also a little bit in your stomach, actually. There are a few of these sensory cells. We can detect sweet, sour, salty, bitter, and umami. That's the one that's been detected most recently. There is actually a separate receptor for umami, and it specifically detects amino acids. So it responds to meaty tastes, and it has its own receptor. That was discovered by a Japanese scientist in the 1990s, although it was postulated for a while. And people suspect that there may be a 6th one for detecting fat. But that one hasn't been proven yet, or we don't have a receptor that's been detected yet either. And it's not true that they're in specific places on the tongue. Probably everybody has seen the little map of the tongue, or supposedly sweet is in here and sour is here, but no, they're all distributed all over the tongue. I don't know who came up with that map and why it's so popular, but it's not actually true that they're localized like that. Any place on the tongue could detect any of those. So this is a slice through one of those taste buds, and you can see the little cells here along the side that are going to actually detect the different molecules. So the taste bud is that little pocket. So here's the papillae. You can see these on your tongue. If you stick your tongue out, they're not microscopic. And then there's little cavities next to those papillae. That's where the taste buds are. And one taste bud, so let me go back for a second. So that's what these are. These are the little taste buds. So this is just a microscope image, and this is a cartoon of the exact same thing. And these individual receptor cells, each one will only respond to specific chemicals in your food and then send a signal. And we can also detect similar things. We also have thermal receptors in our mouth, pain receptors in our mouth, as well as taste buds. Olfaction, so this is things dissolved in the air. This is a part of our experience of food that we eat, because food that you eat, some of those molecules also go up into your nose, and that's part of the flavor of the food, but it's a separate sense. And we have, again, specific receptors that detect certain molecules and send action potentials when they detect those molecules. We don't have a very good sense of smell. Of the 1,200 different olfactory receptor genes that have been identified in mammals, we only have about 380 of them. The rest of the genes, they're there in our genome, but they're mutated and inactivated. They're what we call pseudogenes. So that's why, so there are molecules that other animals like your dog can smell that are in those 1,200 receptor genes that we just literally cannot smell them. They're there, we can't smell them because we have better vision and not as good a sense of smell. Mice as well have all 1,200 olfactory receptor genes are still active. So the reception area is up here in your nasal cavity. Different molecules end up there, whether they're coming through your nose or up through the back. There's also a connection to your nose, to your nasal cavity, through the back of your throat. So when you're eating food, that's where these molecules, how these molecules are getting up into your nasal cavity from the back of your mouth. And they go up there, and if they find a receptor, they bind, and then a signal is sent. And going to the olfactory bulb of the brain. And these olfactory receptor cells, each cell only has one kind of receptor. So there are hundreds of these, or I should more than that, thousands of these olfactory receptor cells that can detect different molecules. And if you get a cold, it has to be wet, because of course these are living cells on the surface. They can't be dry. So there's a mucus layer there that helps keep these cells lubricated and wet. However, too much mucus blocks them. If there's inflammation in this epithelial layer, this is an epithelial layer, it can swell up, have inflammation. That's going to stop the signal from being transmitted, which is why you can temporarily lose your sense of smell if you have a cold or allergies. So we talked about the steps of sensory perception. So the cell or the neuron directly detecting whatever it is, temperature, a molecule, that signal then is transduced into an action potential. It goes from whatever, deformation of a cilia to an action potential. That's transduction. Transduction means something is changed from one form to another. Transmission is that signal now going to the central nervous system. That's transmission. It's action potentials moving from the site of detection to the central nervous system. And then perception is what your brain does with that. Perception is your brain getting an action potential and saying, oh, that's a pain neuron in my mouth. That hurts. Something is injuring my mouth. So perception is what the brain does. Receptors can be specific cells, like the hair cells in the cochlea, or they can be naked neuron dendrites, like the pressure sensors in our skin, or nociceptors in our skin are naked dendrites. Mechanoreceptors detect pressure or deformation. So the cell, like if it's a cilia, the cilia is being moved. That's a mechanoreceptor. Chemoreceptors have receptors for specific molecules, or they can detect pH or osmolarity. Those would be chemoreceptors. Electromagnetic receptors, those would include the receptors, the photoreceptors in our eyes. Thermoreceptors, this would be for both hot and cold, detecting a different temperature than our body temperature. That would be a thermoreceptor. And pain receptors, also called nociceptors, they are detecting high intensity stimulation to any of the other kinds of receptors, really. Things that may injure the body, high amounts of pressure, high heat. Also, they can become much more sensitive and likely to fire an action potential if there's inflammation, prostaglandins around, for example. Hearing and equilibrium, we talked about both of those. And the three different parts of the ear: outer ear, middle ear, inner ear. And it's actually the inner ear where the hearing detection, where the sensory receptors are for both balance and equilibrium and hearing are. Rods and cones are the actual sensory receptor cells in our eyes. Rods are much more sensitive to light, but they can't detect color. Cones are much better at detecting, are the only ones that can detect color, and they're also much better at giving us a fine, detailed picture of something, like for reading. We have taste buds for just 5 tastes in our tongue: sweet, sour, bitter, salty, and umami. And olfaction is everything else that we think is part of flavor or smell or taste that's not in our tongue. And weirdly, we can detect, well, at least it's been shown for sweetness. We can detect sweetness. We have the same exact sensory cells that detect sweetness in our tongue, also in our stomach. We can't really consciously taste things with our stomach. Your stomach is still doing something with that information. Okay, so that's the end of sensory information. The stuff at the end of this chapter about motor control, we're not going to go into that just because of time. So don't worry about that last bit in chapter 50. Okay, that is the end of module 4.